Okay, so welcome to this next video in the playlist on experimental techniques. In this video what we're going to talk about is the Western blotting technique, okay, which is a way of assessing the presence of a certain protein within a sample of proteins. Okay, so let me show you this. So, the Western blot. Okay, right. So basically what we start off with is a solution that may contain a huge number of different proteins. So maybe we start off with a cell lysate. So we have taken cells, we have homogenized them, broken them up to make a lysate, and then we have spun it around and around and around to remove the membranes, basically the insoluble cell membranes, so that what we're left with is the stuff other than lipids, basically. So we're left with proteins, for instance, and that's what's known as a cell lysate. So maybe we start off with a cell lysate or some other solution that contains a huge number of different proteins. Okay, so to illustrate this point, let's have several different proteins within our solution. Okay, so we'll have our beaker here, like so, and it will have many different types of protein here. Let's call this protein A. Okay, we'll have a triangle which can be protein B, and then we'll have a circle which can be protein C. So there are many different proteins present within this sample. Okay, so we'll colour in protein A in turquoise here, we'll colour in protein B in pink, and we'll colour in protein C in blue. And let's say we want to see whether protein A is present within this sample. Okay, Western blotting is a technique for doing that, and it also gives you some idea of how much of the protein that you have in, at least in relative sense. Okay, so if you have one solution that contains twice as much protein A as the other, basically Western blotting can reveal that. It can tell you that one has twice as much as the other. It can't really give you an absolute value for the protein concentration, but it can give you a relative idea of the relative protein concentrations between different samples. Okay, right. So we're going to start off then. We're going to start off with our cell lysate, and basically Western blotting has two components. It has an SDS page portion where we're going to separate out the different members, the different proteins within our cell lysate, and it also has a immunoblotting portion where we're going to use antibodies to detect protein A. Okay, so we're going to start off by putting our cell lysate onto our gel, basically. And in order to do this, what we firstly need to do is add loading buffer, and we need to denature our proteins within our cell lysate. Okay, so basically we're going to start by taking our cell lysate and adding to it loading buffer. Okay, so we're going to put loading buffer in with the cell lysate. Now, what is in loading buffer? Well, there are many different things in loading buffer, but two of the key components that are within loading buffer are bromophenol blue, which is a blue dye, which is why when you see loading buffer, you'll generally find that it's very blue. Okay, and you will also find either SDS or LDS. Okay, so SDS is the original one, and it's the one after which the entire technique is named, but a more modern one, the one that's generally actually used now, is LDS. So let me tell you what these two things stand for. So SDS stands for sodium dodecyl sulfate. So the first S is for sodium, the D is for dodecyl, and then the second S is for sulfate. Okay, and then LDS stands for lithium dodecyl sulfate, so it's pretty much the same thing, but instead of having sodium you have lithium. So let me show you the structure of sodium and lithium dodecyl sulfate, and pretty much they're going to be identical. All that's going to change is which cation we have. Okay, but the dodecyl sulfate anion itself will remain identical. So we'll start off with the dodecyl sulfate, because basically both of these are ionic compounds. You have sodium cations or lithium cations, which I'll have in green here, and then those are bound ionically by electrostatic forces to a dodecyl sulfate anion. So let's discuss the structure of the dodecyl sulfate anion, and then we can just say, and then you'll have a cation bound to this. So dodecyl sulfate basically means that you've got a 12 carbon molecule with a sulfate group right on the end. Okay, so let me draw the structure of one of these out for you. So basically, we'll draw the skeletal structure. So we need 12 carbons. Okay, so we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. That's 12 carbons now, and then this 12 carbon will then be linked to a sulfate group, and the structure of a sulfate group is an oxygen with a sulfur atom in the middle, doubly bound to 2 oxygens like so, and then another oxygen here which has a negative charge on it, and this will then be bound to the cation. So this here, basically, which I've drawn out now, is the dodecyl sulfate anion. Okay, because we've got 12 carbons, which is the dodecyl, a 12 carbon alkane, and then we've got the sulfate group right on the end, and the sulfate group has a negative charge, so this is an anion. Now it can associate then with a cation because this negative charge likes to interact with positively charged species, and that cation can either be a sodium cation to create sodium dodecyl sulfate, or it could be a lithium cation to create lithium dodecyl sulfate. So here is the bit in green, the cation, and then the dodecyl sulfate anion, which is the portion in blue, is all of this. Okay, right. So in the loading buffer you have this sodium dodecyl sulfate or lithium dodecyl sulfate. Now at present you can't see what this is going to do yet, so I'm going to ask you to take it on faith that you add a loading buffer which contains these two, and we will come back to what they do. We'll come back to what SDS or LDS is going to do very shortly, but the bromophenol blue will take a little longer to get to why we put that in. So at the moment all we've done is we've taken our mixture of proteins and we've added in loading buffer. Okay, now basically you have a certain sized well that you are going to fill, basically, and these wells accommodate 20 microliters generally, so the loading volume is generally around 20 microliters. Okay, so you need your samples that you're putting in, which will contain the cell lysate, the loading buffer, and also one other thing, which is your reducing agent, which I'll come to in a moment. You need that to have a volume of 20 microliters, because when you load it onto the gel, okay, so when you stick your sample into the little wells in the gel, which we'll come to in a moment, the well will only accommodate around 20 microliters. Okay, so we want to produce samples which have got a volume of 20 microliters. Now, loading buffer usually comes in what is known as 4 times loading buffer. So what you'll generally find is that if you go to the shelves, you'll find something called 4 times loading buffer. Now what that means is that this is 4 times more concentrated than it should be, basically. So when you actually are using this in a sample which you're about to load onto a polyacrylamide gel, you don't want it to be this concentrated. You want it to be 4 times less concentrated. Now what does that actually mean? Well, if, for instance, we want a loading volume of 20 microliters, if we want to put in 20 microliters of fluid into the well, then that means that a quarter of that needs to be the loading buffer. So 5 microliters of this needs to be the loading buffer. Okay, so that's what 4 times means. It means that it's 4 times too concentrated. So a quarter of the loading volume that you're going to use needs to be devoted to this, and that will mean then that when this 5 microliters of loading buffer is diluted into the 20 microliters that we're going to make the whole sample which we're going to load, it will be the correct concentration. It'll be 1/4 times the concentration of the loading buffer that's stored in the glass bottle on the shelves. Okay, and that means that the concentration of the loading buffer will be properly correct in our sample. Okay, so we've got another 15 microliters to fill. Now, we've added loading buffer. We haven't yet added our cell lysate. We'll do that last. There's one more thing that we're going to need to be in our samples that we're going to load onto the polyacrylamide gel. Okay, and this is what's known as a reducing agent. Okay, and we'll come back to the function of this in a moment. So there are 2 reducing agents which you can use, basically, well, which are commonly used. And these are DTT and beta-mercaptoethanol. So we're going to discuss DTT, but we'll also discuss beta-mercaptoethanol because DTT is more commonly used. It's a nicer one than beta-mercaptoethanol. So DTT, basically, if we have a look at its structure, what its structure looks like so. It's basically a 4-carbon molecule, so 1, 2, 3, 4, and I'm sorry about that, that's going to be a thiol group here, so a thiol group off here, and you've got 2 thiol groups coming off this 4-carbon structure. Okay, now, I haven't actually told you what DTT stands for. So DTT stands for dithiofriatol. Okay, dithiofriatol. Okay, so basically you have 2 thiol groups off this 4-carbon molecule, but that's not all you have. You also have 2 alcohol groups coming off this 4-carbon molecule, one here and one here. So it's nicely symmetric. You've got this 4-carbon backbone, 2 alcohol groups coming off the 2nd and the 3rd carbon, and then off the 1st and the 4th carbon you then have thiol groups, and this is known as dithiofriatol. Okay, and abbreviated, it's then D for di, T for thio, and then the second T is for friatol. Right, so dithiofriatol is one reducing agent, okay, and this means that it's going to be able to break disulfide bonds, and this is going to become very important because Western blotting is going to contain it usually contains a denaturing polyacrylamide gel electrophoresis, okay, and that's the sort that we're going to look at. We're going to look at denaturing gel electrophoresis within the Western blotting, and in order to denature the proteins, we need to break all tertiary bonds. We want it to return to a completely linear polypeptide. Okay, and part of breaking all of those tertiary bonds means that we're going to have to break the disulfide bonds. Now, disulfide bonds will not break under temperature, at least not any sort of temperature that you would want to use. Okay, so you need a chemical agent that's going to be able to break them, and reducing agents are going to be able to do that. And you'll notice that they have thiol groups, just like cysteine residues, so it seems hopeful that they're going to be able to bind together and donate protons away, which will then go on to the disulfide bond to reduce them then back to thiol groups. Okay, the other reducing agent that can be used is beta-mercaptoethanol, which is sometimes abbreviated to BME. So this stands for beta-mercaptoethanol. Okay, so let me show you now the structure of beta-mercaptoethanol. Okay, so basically, beta-mercapto tells you that you've just got a thiol group basically off the 2nd carbon. Then we've got ethanol. So basically, all we need to draw is a 2-carbon molecule like so. You then have an alcohol group, that gives you ethanol. Okay, and now you want a thiol group, and the other name for the old name for thiol groups is mercapto. So that just means you have a thiol group coming off the beta carbon, because you'd view the alpha carbon as the one with the alcohol group on, and then the beta one as the second one. Okay, so beta-mercaptoethanol, and that still often is called beta-mercaptoethanol. You can use beta-mercaptoethanol as a reducing agent, but it's very, very strongly smelling, and it's not particularly pleasant to use. So people generally use dithiofriatol.